Biology, at every level, is a tangle of nonlinear systems. Organs do not simply add up their parts. Cells do not behave like predictable machines. Molecules in a reaction network often amplify or dampen each other in ways that defy straight linear logic. To decode life, scientists must wrestle with this non-linearity, the property that small inputs can cause huge effects and feedback loops can generate rhythms, switches, or chaos. Take the human heart. Its pumping is not a simple piston but a self-regulating oscillator driven by networks of excitable cells. A tiny disruption in the ion currents of just a few cells can cascade into an arrhythmia that threatens the entire organ. Or consider the brain, where billions of neurons interact nonlinearly. A single spike in one pathway can shift mood, memory, or decision-making, outcomes far larger than the spark that triggered them. Doesn't it feel almost paradoxical that stability in such systems arises not from linear order, but from dynamic feedback among countless unstable parts? Zoom down to the cellular level, and nonlinearity becomes even more pronounced. Signaling pathways often contain positive feedback loops that act as switches, turning a cell on or off irreversibly once a threshold is crossed. Other networks use negative feedback to maintain balance, like thermostat circuits. For example, insulin and glucose interact in a nonlinear dance. Too much sugar spikes insulin release, which then overshoots, sometimes driving the system into oscillations. Could this explain why metabolism is so sensitive and why small disruptions can tip into diseases like diabetes? At the molecular scale, chemical reactions themselves are nonlinear. Enzymes can act cooperatively, where binding of one molecule makes the next bind faster, a principle underlying hemoglobin's oxygen transport. Oscillating reactions like the Belousov-Jabotinsky reaction demonstrate how feedback can turn chemistry into a clock, pulsing in colorful waves. In living cells, similar nonlinear chemistry drives circadian rhythms and cell cycle progression. Without these built-in oscillators, life could not maintain order against constant noise. The scientific challenge is that nonlinear systems are notoriously hard to predict. Linear equations give neat solutions. Nonlinear ones often require simulations, approximations, or entirely new frameworks like chaos theory, network analysis, or stochastic thermodynamics. Yet, these very methods have revealed hidden order, strange attractors, robustness in noisy circuits, and emergent properties where the whole is more than the sum of its parts. Isn't it fascinating that what looks like chaos may actually be structured at a deeper level? Understanding nonlinear biology has profound implications. It means diseases are not just local defects, but network instabilities. Therapies might need to nudge whole systems, not just fix a part. It means that evolution has harnessed nonlinear feedback to create robustness out of fragility. And it suggests that life itself, oscillating between order and chaos, may be the ultimate example of how nonlinear dynamics generate complexity. Biological organs such as the heart, lungs, and brain do not operate in a strictly proportional manner, but instead respond in highly nonlinear ways where small changes in inputs can yield disproportionately large or unexpected outputs. For instance, the heart can remain stable under gradual increases in workload, but may suddenly transition into arrhythmia once a hidden threshold is crossed. These behaviors arise because organs consist of vast networks of interacting cells, feedback loops, and oscillatory processes that cannot be reduced to simple cause and effect chains. Nonlinear dynamics allow organs to be both robust against minor disturbances and yet capable of rapid state changes when pushed beyond critical points. This duality makes organs resilient in normal conditions, but also vulnerable to abrupt failures under stress. Understanding these properties requires combining physiology with mathematical models of bifurcation and chaos theory. A healthy heart maintains rhythmic beating, but under stress, it can suddenly enter fibrillation without gradual warning. It is like a suspension bridge that sways gently under light winds, but may abruptly collapse when wind speed crosses a critical threshold. Tissues behave as emergent systems where the behavior of individual cells aggregates into collective dynamics that cannot be predicted by studying one cell in isolation. Mechanical coupling electrical signaling, and chemical diffusion create feedback loops where local changes propagate non-linearly across the tissue. These collective effects produce phenomena such as synchronized beating in cardiac tissue, or wave-like calcium signaling in epithelia. Nonlinear interactions also enable tissues to self-organize, repair damage, and adapt to fluctuating environments, which are essential properties of living systems. However, the same nonlinear rules can also generate pathological states such as tumors, where growth becomes uncontrollable once signaling pathways surpass certain thresholds. Thus, tissues embody both the creativity and the fragility of nonlinear biological dynamics. In cardiac tissue, a few misfiring cells can trigger large-scale arrhythmias that disrupt the entire organ. It is like a stadium crowd where a single chant can rapidly spread and amplify into a deafening roar. At the cellular level, nonlinear systems govern decisions such as whether a cell divides, differentiates, or undergoes programmed death. Molecular switches and feedback loops amplify small biochemical signals into decisive outcomes, preventing cells from remaining in indecisive intermediate states. These dynamics often involve threshold effects, bistability, and hysteresis, meaning that past states influence future choices. Such nonlinear control ensures that cell fate decisions are both robust and irreversible once committed. Importantly, noise in molecular interactions does not merely disrupt these decisions, but can act as a trigger that nudges the system across thresholds. Cells, therefore, exploit nonlinear mechanisms to transform microscopic randomness into macroscopic reliability. A stem cell exposed to slightly different concentrations of growth factors may either remain undifferentiated or suddenly commit to a specialized lineage. It is like a thermostat that keeps a room steady until the temperature crosses a threshold, after which the heater or cooler rapidly switches on. Chemical reactions in biology often show nonlinear behavior, especially when feedback or autocatalysis is involved. Enzyme kinetics can exhibit saturation, cooperative binding, or oscillations rather than smooth proportionality. These effects allow small fluctuations in molecular concentration to trigger sharp transitions in reaction rates, creating switch-like responses. In metabolic pathways, such nonlinearities are crucial for regulating energy flow, preventing futile cycles, and coordinating multiple processes simultaneously. They also explain why biological systems can respond rapidly to environmental cues, even when molecular concentrations are tiny. Without nonlinear kinetics, life would lack the responsiveness and adaptability required to survive in dynamic environments. Hemoglobin binds oxygen cooperatively, where binding of one oxygen molecule sharply increases affinity for others. It is like a zipper that resists movement at first, but then closes quickly once a few teeth are aligned. At microscopic scales, noise and fluctuations are not just disturbances, but active participants in nonlinear biological processes. Random thermal motion can push molecules or cells across hidden thresholds, thereby triggering switches or oscillations. Nonlinear systems amplify these small fluctuations into macroscopically significant outcomes, such as rhythmic patterns or synchronized activity. This stochastic resonance transforms randomness into order, making noise a functional part of biological dynamics. Far from equilibrium, living systems are constantly balancing noise with feedback to achieve adaptive responses. Thus, noise is not merely tolerated, but recruited as an integral element of cellular computation. Random fluctuations in gene expression can cause genetically identical bacteria to follow different survival strategies. It is like the flutter of leaves in the wind, seeding large swirls and eddies in the forest air. Nonlinear systems in biology rely heavily on feedback loops, which can stabilize or destabilize processes depending on their sign and strength. Negative feedback ensures homeostasis, while positive feedback can amplify signals into irreversible outcomes. In complex networks, multiple feedback loops interact, producing oscillations, bistability, or chaos. These interactions are essential for processes like circadian rhythms, immune responses, and developmental pathways. The balance between feedback types determines whether a system resists change, amplifies it, or oscillates between states. Therefore, feedback is the architecture through which nonlinearity becomes functional. The circadian clock relies on transcriptional feedback loops that generate tilde 24-hour oscillations. It is like a microphone near a speaker. Carefully tuned, it stabilizes sound, but slightly misaligned, it produces runaway feedback. Living systems persist by maintaining themselves far from equilibrium through constant energy dissipation. Non-linear processes, such as chemical oscillations and molecular motors, exploit this disequilibrium to generate order and perform work. The second law of thermodynamics still holds, but life achieves local decreases in entropy by exporting disorder to the environment. These dynamics explain how self-organization, pattern formation, and adaptability are possible in living matter. Non-equilibrium thermodynamics therefore provides a unifying framework for understanding non-linear biological dynamics across scales. Without this constant energy flow, biological systems would decay into equilibrium and lose their complex structures. Cells consume ATP continuously to maintain ion gradients across membranes, which would otherwise collapse. It is like a refrigerator that maintains cool order inside by constantly expelling heat outside. Scientists struggle to probe nonlinear systems because classical instruments are designed for linear cause and effect measurements. Capturing feedback, thresholds, and stochastic amplification requires dynamic, high-resolution, and high-throughput tools. Conventional averaging techniques often erase nonlinear phenomena by smoothing out variability. Emerging methods such as single-molecule microscopy, microfluidic gradient chips, and real-time biosensors now allow glimpses of nonlinear behavior in action. However, these technologies are still limited in resolution, speed, and ability to track multi-scale interactions simultaneously. The challenge is building instruments that can match the complexity of the biological systems they seek to measure. Single-molecule fluorescence microscopy reveals bursts of enzyme activity that average measurements would have hidden. It is like hearing only the overall murmur of a crowd until a directional microphone lets you isolate individual voices. Decoding nonlinear systems has profound implications for diagnosing, predicting, and controlling disease. Many pathologies, from epilepsy to cancer, reflect failures of nonlinear control, where thresholds are crossed or feedback loops destabilize. Understanding these dynamics enables targeted interventions that restore stability or exploit tipping points. In biotechnology, nonlinear design principles guide the creation of synthetic circuits that can oscillate, switch, or self-organize in useful ways. Nonlinear models also inform personalized medicine by predicting when small differences in patients' parameters may lead to dramatically different outcomes. Thus, mastering nonlinear biology is key to both healing disease and engineering life. Cancer therapies increasingly target feedback pathways in signaling networks rather than single genes. It is like fixing traffic jams not by widening one road, but by redesigning the whole network of intersections.